We're going to start off this section with a quick reminder of a reaction we did in Chapter 15. When we have an aldehyde or a ketone, they can react with alcohols to form hemiacetals. They may also react with additional alcohols to become acetals. We talked about this reaction or these reactions in Chapter 15 because carbohydrates always have both of these pieces. And the remainder of the chapter is about either forming hemiacetals and monosaccharides or joining them together as acetals and polysaccharides. Whenever we dissolve a hexose or pentose in water, it has a tendency to react with itself. One of the alcohols will react with the aldehyde or ketone from the same molecule. These can form a wide variety of cyclic, meaning ring, hemiacetals, which are mostly either 5-membered rings, which we call furanoses, and 6-membered rings that we call pyranoses. We always will draw furanoses in the same orientation. The oxygen is going to be on the top of that pentagon, and the hemiacetal carbon is always going to be immediately clockwise from the oxygen. We also draw pyranoses the same way every time. The oxygen will be on the top right of the hexagon, and the hemiacetal will be immediately clockwise from the oxygen. On many carbohydrates, we may have an alcohol that gives us a furanose, and an alcohol next to it that would give us a pyranose. But for all the carbohydrates that we'll look at in this course, only one of those is really going to be important. The rings that form will always be between the carbonyl carbon and the second to last alcohol's oxygen. If we want to figure out if the linear monosaccharide is a furanose or a pyranose when we form the ring, we just count the atoms from the carbonyl up to the oxygen that it is bound with, including the carbonyl carbon and the oxygen. For this molecule, which is D-glucose, there are 6 atoms total, starting again from the carbonyl and going to the oxygen. And so this will become a pyranose when it forms a hemiacetal. If you couldn't count 6, make sure that you pause and take time to make sure you find out where those 6 come from. The next molecule, D-fructose, is a ketose. So its carbonyl is lower at the 2 position instead of at the 1 position, like in an aldose. The second to last alcohol's oxygen is here. And this, if we count the carbon all the way up to the oxygen, only gives us 5 atoms that would be in the ring, so a furanose. Look at the last molecule, which is D-ribose. Which atoms will react in the last molecule? The carbonyl and the second to last alcohol react just as in the other examples. Now, based on this, will it be a furanose or a pyranose? It should be a furanose since there are only 5 atoms, including the carbonyl and the oxygen. So what actually happens when we form a cyclic monosaccharide? Remember, the Fischer projection is drawn linearly, but it really represents a molecule that's arranged in a ring with all the carbons angled back. This is a helpful way of thinking about the structure because it generally matches what happens when we form a ring. So a lot of the structure is already there in the Fischer projection. Let's walk through what's going on in this picture in order. The Fischer projection shown at step number 1 up here actually represents the form of the molecule that's shown in 2. We already know that glucose will form a pyranose since there are 6 atoms from the carbonyl to the last chiral oxygen down here. We want to make sure that aldehyde is where the hemiacetal will be. So when we draw out a pyranose like this, we'd have the hemiacetal immediately clockwise from the oxygen in the pyranose, or 3 o'clock in the ring. Notice that when we arrange all the carbons as we do in number 2, everything perfectly matches the Fischer projection, since all the carbons are pointed back. Every oxygen that was on the right in the Fischer projection now ends up down in the ring, and every oxygen that was on the left ends up in the ring in this form. However, the form that we see in number 2 puts the 6th and final carbon of the chain right here, right next to the aldehyde. But that carbon's not actually what interacts with the aldehyde. What interacts with the aldehyde is the alcohol that's on the 5th carbon, so right here. And so we need to rotate that into the ring for it to be close enough. This is the change that's occurring between 2 and 3. We're rotating that OH up. Notice that because the oxygen that was pointed down needed to be rotated up to be part of the ring, the carbon that was there, that carbon number 6, rotates up to make room for it. When the alcohol is in the ring, it can form a hemiacetal with aldehyde at position 1 in the ring. This means that the direction of the alcohol on carbons 2, 3, and 4 all directly correlate to the Fisher projection. So if we look at this, on carbon 2 and 4, we have the OH on the right, and those will correspond to the OH being down in the ring. And when we look at the oxygen here on 3, which is on the left, that correlates with the oxygen being up on carbon number 3 in the ring. The direction of the CH2OH that is attached to carbon 5, so the 6th carbon of this chain, is now actually going to be different, because we're not looking at an oxygen, we're looking at a carbon. That carbon had to be rotated up to make room for the oxygen that was pointed down. So for all the oxygens that are on the right, like we see in the Fisher projection, we'll end up with the final CH2OH being pointed up, the opposite of if it was the oxygen itself. Notice that the oxygen that's actually forming the hemiacetal in this reaction is the one that defines whether we have a D or L carbohydrate. Since D carbohydrates are by far the most commonly occurring naturally, the OH being on the right is also the most common, and therefore the ring with the CH2 pointed up is also the most common form naturally. At this point, step 4 in this diagram, we can explain the direction of all the bonds from carbons 2 to 5, matching up with the Fisher projection here. Carbon 6 isn't chiral, so we don't need to worry about the directions of its bonds. That leaves just carbon 1, the aldehyde that eventually becomes a hemiacetal. Since the aldehyde is not chiral, we actually can make 2 versions of this ring. The one that's shown up here, the alpha D glucose, will have the OH of the hemiacetal on the opposite side of the CH2OH. Again, we say this is the alpha version. Alpha has the hemiacetal OH on the opposite side of the ring than the CH2OH. We can also make a version of this molecule with the OH pointed on the same side as the CH2OH. So here we have the OH and the CH2OH both on the top of the ring. We call this the beta version of the ring, so hence beta D glucose. The alpha and beta versions are different only at this carbon, which makes them epimers. Again, if you don't remember the term epimers, that's diastereomers that are different at just one carbon. The alpha form and beta form can be converted back and forth pretty much constantly, and they're constantly converting between alpha and beta form. Even though the hemiacetal breaks open to the aldehyde when it's being converted back and forth, the aldehyde form, which we call the open chain, is very quickly closed again. So both the alpha and beta forms of glucose have distinct properties, but they're quickly and constantly converted back and forth. And because the 2 epimeric forms are so quickly and easily interconverted, we call them anomers, a specific term for the carbohydrate epimer chiral center that's quickly interconverted. So anomers are a specific type of epimer that happens here with the hemiacetals. Again, the anomeric carbon, that's again the one that can have the OH pointed up or down, is the hemiacetal carbon. And it's always in the same place on these molecules, immediately clockwise from the oxygen in the ring. It can change because it's not chiral in the aldehyde form and only becomes chiral when it becomes a hemiacetal. We typically draw cycles or rings as Haworth projections that follow a set of standard formatting rules to make them easy to identify. We've already identified that the basic structure of the ring, either pyranose or furanose, relates back to the number of atoms from the carbonyl, the second to last carbon's oxygen. We can trace this backwards to figure out what kind of monosaccharide is represented too. The first thing that we want to do is identify the hemiacetal. This is convenient in Haworth projections because the hemiacetal is always immediately clockwise in the ring from the oxygen. This hemiacetal can either only be connected to carbons of the ring, which would make it primary. There's only carbons in one direction. And then it would be related to an aldehyde or aldose. This is the case in this molecule. If it were a ketose related to a ketone, it would need another carbon attached outside the ring, since ketones always have two carbons attached to the ketone carbon. We can see an example like that in the far right. So we already know that since the hemiacetal is primary, the carbohydrate is an aldose. We can also count exactly how many carbons are in the molecule. Since the leftmost Haworth projection has 6 carbons, we know that the carbohydrate is a hexose. The direction of the CH2OH counterclockwise from the O in the ring always corresponds with whether the carbohydrate direction is D or L. A CH2OH pointed up is the most common and corresponds to the more common D carbohydrates, which have the last chiral carbons OH on the right. We can also tell what anomer we have, either alpha or beta, based on the direction of the hemiacetal OH. If the hemiacetal OH, down here, is opposite the CH2OH we just looked at, we have the alpha anomer. If it was on the same side as the CH2OH that we just looked at, it would be beta. Now look at the next molecule and try to identify whether it's an aldose or a ketose. Since the hemiacetal carbon is primary, that is just attached to one carbon, we know that it's an aldose. Since it is 6 carbons long, we know it's a hexose. And since the CH2OH counterclockwise from the ring oxygen is pointed up, we know that we have a D carbohydrate. And finally, when we compare the hemiacetal alcohol to that CH2OH that we just looked at, we see now that they're both pointed up, making this a beta anomer. Now try to identify these things for the final molecule. First, the hemiacetal is attached to the next carbon in the ring, but also a carbon outside the ring, making it secondary, a ketose. It has 6 carbons total, making it a hexose, and the CH2OH that isn't on the hemiacetal is pointed up. It's a D carbohydrate. And finally, it's opposite of the OH of the hemiacetal, so it's the alpha anomer. Notice that the direction of the CH2OH on the hemiacetal is determined by the direction of the OH. Right, so when we're looking at these pieces, this goes up because this is already down. The information that we've put together from the hemiacetal so far can help us to convert back to the first structure. First, if we know that it's an aldose, we can draw the aldehyde. If we know that there are 6 carbons, we can draw 5 carbons below that. Below the carbonyl in the Fischer projection, every carbon will be chiral except for that final carbon. So the remaining 5 carbons give us 4 chiral centers and 1 carbon that isn't chiral at the very end. The final chiral center is always the one that determines D or L. We know that it's a D carbohydrate, so we know that that oxygen is on the right. This gives us most of the Fischer projection already. The remaining pieces are filled in based on the 3 carbons between the hemiacetal and the carbon with the CH2OH on it. So we're looking at this carbon, and this carbon. Notice that since this is the hemiacetal and matches up with this, we're looking at now these 3 carbons as well, the ones that aren't marked. The OHs that are on the top here, like at position 2 and position 4, correspond to left-pointing OHs in the Fischer projection, and the OHs on the right correspond to OHs that are OHs on the pointed down correspond to OHs on the right. So notice that the order that we just filled in, or just are about to fill in, was left, right, left. It starts with a left-pointing or up-pointing group. The first OH pointed down is actually part of the hemiacetal. Again, we're not looking at this. We're looking at only these 3 carbons. Now you try to draw out the Fischer projections for the remaining 2 molecules. The next molecule is a D-aldohexose, just like the last molecule. It should have 4 chiral centers with the last chiral carbon's alcohol on the right because it's D. On carbons 2 through 4, the other three chiral centers, the Haworth projection has alcohols down, up, then down, corresponding to right, left, and then right again in the Fischer projection. But again, we're only looking at these three chiral centers corresponding to these three chiral centers on the Haworth projection. The hemiacetal isn't chiral once we move it to the Fischer projection because it's an aldehyde. And this carbon follows different rules because it's a carbon and not an OH. It's actually this carbon pointed down. Now try the last one. This molecule is a D-ketohexose, meaning the first chiral carbon, or first carbon isn't chiral, and the second carbon is a ketone. So we have 1, 2 carbons before we start to hit actual chiral centers, corresponding with 1, 2 down here. Only the alcohols on carbons 3 and 4, correspond directly to the Fischer projection. In this case, they're both drawn down in the ring, so they'll both be right in the Fischer projection. And the final chiral carbon has the OH on the right because this is a D-carbohydrate, which we see with the CH2OH up here being on the left. To go from Fischer projection to Haworth projection, we start by identifying whether a carbohydrate is D or L and whether we have a pyranose or a furanose. Since the last chiral carbon has the OH on the right in this first example, we know that the molecule is a D-carbohydrate. We can also tell that it's a pyranose because there are 6 atoms from the carbonyl, being 1, to the last chiral carbon's OH, being number 6. A D-pyranose will have this basic structure. A 6-membered ring with a CH2OH sticking up out of the ring just to the left of the oxygen in the ring. That corresponds with the D-carbohydrate piece. Once we have the carbon sticking out of the ring, we can put the hemiacetal OH in the right direction. This information doesn't actually come from the Fischer projection. It needs to be given as either alpha or beta. Otherwise, either direction is equally correct for the hemiacetal, because the aldehyde could react to form either form. For these examples, I just put alpha or beta directly below the Fischer projection in order to indicate which one we want to do. The alpha enomer will have the hemiacetal OH opposite the CH2OH we just drew, meaning it will look like this. It's important to do the D-carbohydrate carbon first, since the designation alpha or beta for the hemiacetal alcohol is always relative to the CH2OH on the other side of the ring, oxygen. So we have to do the D because it determines what direction the alpha or beta will actually be. I would recommend also doing the hemiacetal carbon before any of the middle carbons. It's really tempting to treat the hemiacetal, which again corresponds to the aldehyde or ketone, as the first chiral carbon in the Fischer projection. But the hemiacetal corresponds with the aldehyde, not with the other piece. So this and this correspond together, where the chiral carbons here correspond to these carbons. Mixing those up is a common mistake, and it results in all of your carbons being shifted by 1. So again, if you do the hemiacetal first, it's easier to keep track of which carbons are already accounted for because we already have the OH assigned before we look at the chirality on carbons 2 to 4, the ones highlighted in green. Anyway, in this case, they are right, left, and right in the Fischer projection, corresponding to down, up, down in the Haworth projection. This, by the way, is alpha-D-glucose. Using the same pattern, try to draw out the next two. If you're stuck, first start with D or L. Which is it? And will it be a pyranose, which would be if you have 6 atoms in the ring, or a furanose if you have 5 atoms in the ring? So a D-furanose looks like this to start with. And we see that we're looking for the beta anomer in this problem from the beta bottom right of the Fischer projection itself. This beta means that the hemiacetal OH will be on the same side, that is the top, of this ring. as is the CH2OH that isn't on the hemiacetal. If we put the OH up top, the CH2OH that's attached to the hemiacetal, this one down here, needs to go to the bottom since the OH is already up. At this point, we can just fill in the remaining two chiral centers, this one and this one, which correspond to here and here. Left-right in the Fisher projection gives up and then down in the ring as we move clockwise around the ring. The molecule we just drew is beta-D fructose. The two Haworth forms that are shown here, the alpha-D glucose and the beta-D fructose, are the two pieces that will tie together to make a sucrose in a few slides. The final molecule is a D-furanose again, and it's designated alpha. The OHs are all on the right of the Fisher projection, so the carbons that aren't already connected to oxygen are filled in with the OHs pointed down. In addition to forming hemiacetal rings, monosaccharides and even disaccharides, which together we classify as sugars, can go through a few additional reactions and sometimes do in our bodies. Because carbohydrates have alcohols and aldehydes, sugars are often readily oxidized. The most common locations are either at the first or last carbon. The first carbon often being an aldehyde and the last carbon being an alcohol that's quite accessible. Ketoses can isomerize to aldoses under some oxidizing conditions, which is what we see in the Benedict's test in lab, which is why even though we don't expect ketones to oxidize, ketoses can, because they actually become aldehydes first. The aldehyde, or ketones, again through isomerization, are more reactive than alcohols, and so there are many tests for oxidizing only the aldehyde or ketone. So when a carbohydrate is oxidized, it's going to reduce something else, which gives the carbohydrate the name a reducing sugar. Again, this only happens when we have either the open form with an aldehyde or ketone that can isomerize to an aldehyde, or in the hemiacetal form, which can easily open. It never happens if there are no hemiacetals, aldehydes, or ketones, like if everything's an acetal, which is what we'll see in the case really only in very particular disaccharide linkages. The aldehyde or ketone can be reduced back to an alcohol as well, and we'll see some of these products making up components of some important cofactors for central metabolism. So we'll just see some examples of things that used to be carbohydrates. We'll see carbohydrates form phosphoesters as well, which is something that our cells do to help keep them in the cell and make it easier to gain energy from them. We'll talk about that in chapter 24. We'll also see some examples where the second alcohol is swapped for an amine. We won't look at this reaction, but we'll see some polysaccharides that look just a little bit different because this reaction has happened on the monosaccharide components. And finally, we'll also form acetals, which is really the subject of the rest of this chapter. Enzymes in our bodies can link together individual monosaccharides into longer chains by reacting the hemiacetal of one of the monosaccharides with the alcohol from another. So here's the hemiacetal, here it's reacting with the alcohol. While hemiacetals can convert to the linear open chain form pretty readily and are always converting back and forth, this doesn't happen with acetals. Once an acetal is formed, it's relatively permanent until an enzyme breaks down. This means that while the form, alpha or beta, of the monosaccharide was temporary, so this happens to be in the alpha form, the form of an acetal in a disaccharide is more or less permanent, and so this is permanently linked in the alpha form now. Biologists call any bond between a monosaccharide and another molecule a glycoside bond, and so the links between the monosaccharides are also considered glycoside bonds or glycoside linkages, which is just another way of referring to the acetal here. Again, these are always acetals in every example that we'll look at in this course. Because the acetal bonds, or glycoside bonds, are not easily broken, the direction of the oxygen in the acetal, either alpha or beta, will give us different molecules, different properties. Glycoside bonds are defined by two things. First, the position of the acetal or acetals, that's whether they're alpha or beta. And second, which carbons are connected between the monosaccharides. So here we have an example which is maltose, a disaccharide of two glucoses. It has the acetal carbon locked into the alpha position. We know it's alpha because the oxygen outside of the ring is pointed down, and it's on the opposite side of the CH2OH for that ring. The acetal carbon, which is at the 1 position, is connected to the 4 position of the other glucose. So then we'd say that the glycoside bond is alpha 1 to 4. We indicate that with an alpha sign and then a parentheses, 1 arrow 4. We can have many different glycoside bonds between carbohydrates, but alpha 1,4 linkages, like the linkage shown on the top, are very common. This is the major linkage of not just maltose, but also of the polysaccharides starch, or amylose, amylopectin, and glycogen. Notice the way alpha 1,4 is written. The Greek letter alpha, then we have a parentheses that enclose 1 with an arrow to 4. We can also have beta 1,4 linkages, where the acetal O that's outside of the ring is pointed in the same direction as that ring's CH2OH. By only flipping a single chiral center, we have completely changed the properties of this molecule. When glucose is bound beta 1,4, we human folk can't actually digest it. Notice that our drawing of maltose on the left has a prefix alpha as well. So it says alpha maltose. This alpha comes from the remaining hemiacetal on the rightmost glucose, which can still be opened and closed. So this is the hemiacetal that would make maltose a reducing sugar. So alpha maltose can convert back to an open chain with a reactive aldehyde, like we see here, and then it can be converted to beta maltose, and it can always be converted back to alpha maltose. But the whole time, the acetal bond right here, or glycoside bond, stays locked in the alpha 1,4 conformation. Because maltose has this convertible hemiacetal, the aldehyde can react, and again, it's considered a reducing sugar. We'll see this pretty much anytime there's a hemiacetal. The aldehyde, or even a ketone, can react, allowing these carbohydrates to be considered reducing sugars. While maltose joins two glucoses, alpha 1,4, we can also join those two glucoses, beta 1,4. So this disaccharide, called cellobiose, is not digestible in our bodies. Again, it's on the basis of simply flipping the direction of one bond. Only this acetal bond has flipped between these molecules. You should know for each of these disaccharides what components make them up. So again, for maltose and cellobiose, it's glucose in each case, D-glucose, and the name for their glycoside bonds. So for maltose, it's two D-glucose molecules joined by an alpha 1,4 link. For cellobiose, it's two D-glucoses joined by a beta 1,4 link. Not all disaccharides are made up of the same monosaccharide. Here are two disaccharides with different monosaccharides. And again, I'd like you to know the components and how they're linked. So lactose, an important disaccharide in dairy products, is beta-D-galactose linked to glucose in a beta 1,4 link. Sucrose, shown below, has a particularly interesting glycoside link. The glucose is linked in the alpha position. It's locked in there as an acetal, because the OH is opposite the CH2OH on the same ring. But the alcohol that glucose is linking to is actually the alcohol of fructose's own hemiacetal that's locked in the beta form. Since both hemiacetals are linked together as acetals, so we have an acetal here and an acetal here, there's no reducing end, there's no end that's a hemiacetal, and sucrose is considered a non-reducing sugar. But when we hydrolyze sucrose, it converts back to the monosaccharides, glucose, and back to the monosaccharides, fructose, which both have hemiacetals and are therefore reducing. When we name sucrose's linkage between glucose and fructose, we say that we have an alpha comma beta linkage, 1 arrow 2, or alpha beta 1, 2 linkage. We need the alpha because glucose is an alpha acetal, and beta because fructose is a beta acetal. The glucose acetal is at 1 but since fructose is a ketose, its acetal is at 2. With monosaccharides, we saw a huge variety of carbohydrates, really just based on functional group, length, and direction of bonds. When we joined these together into disaccharides, we could vary the sugars that were linked together, or the monosaccharides that were linked together, and we could vary the direction of the links. We can still change all of those things in a polysaccharide, which is also known as a glycan, but we also have a wide variation in length, and we can even have branching if we have alcohols at different places on the same monosaccharide all become acetals. When a polysaccharide is made up of all the same monosaccharide, it's called a homopolysaccharide. We can see those examples on the bottom left, the leftmost 2 examples. When we have different units, it's called a heteropolysaccharide, the rightmost 2 examples. For each of the polysaccharides that we talk about next, I'd like you to have some idea of what it does, what its units are, like glucose versus galactose, and its linkages, like alpha-1,4 or beta-1,4, and finally if it's branched. So listen and take note of each of those things. What carbohydrates are making up what monosaccharides, the linkages, whether it's branched or not, and generally what it does. We have 3 classes of polysaccharides. The first 2, storage polysaccharides and structural polysaccharides, are based on what the polysaccharide does. They either store energy or help give an organism structure. Acidic polysaccharides are named based on modifications to the polysaccharide that make them more acidic, so we'll have specific examples there. Our first type of polysaccharides are energy storage polysaccharides. The simplest energy storage polysaccharide is amylose, or starch. This is a major way that plants store energy. Every unit of amylose is glucose, and every one of those links between the glucoses is alpha-1,4. So this is an unbranched polysaccharide. With one straight chain, it gets up to about 1,000 units of glucose. Not bad. Plants do need a more dense way to store carbohydrates, so they also store them as amylopectin. Amylopectin has long chains of alpha-1,4 linked glucose units. So it's all glucose, it's a homopolysaccharide, and its linkages are mostly alpha-1,4. But it also has occasional branching, where the 6th carbon's alcohol also reacts with glucose, hemiacetal, to form alpha-1,6 linkages. This creates a much more dense structure, and amylopectin can contain about 100,000 glucose units or so. Notice that of all those 100,000 units, only one of them keeps its hemiacetal on the very end of the molecule. Every other unit has the hemiacetal tied to another unit as an acetal. So even where we have branching, we have acetals linking to the next position. Since animals, including us, have to move around and can't stay in one place, we need to store carbohydrates even more densely. We do this by having more branching. Glycogen, the way that we store glucose, is just a more branched and therefore bigger form of amylopectin. It still is all glucose, a homopolysaccharide, with alpha-1,4 and alpha-1,6 units. Our 2 structural polysaccharides, cellulose on top and chitin on the bottom, are both versions of glucose that are linked beta-1,4. Neither are digestible to energy in our bodies, but play an important role as dietary fiber. Cellulose gives plants their structures, while chitin gives insects and fungi structure. Notice that the glucoses that are used in chitin have been modified to become an amino sugar, and then amidified. This is the only structural difference between cellulose and chitin, which are otherwise both unbranched homopolysaccharides with beta-1,4 linkages of glucose units. Our final class of polysaccharides are the acidic polysaccharides. I don't care as much about you knowing the components and links here, but I do want you to notice that each has acidic groups. Hyaluronic acid, which is a joint lubricant, is more soluble than it would otherwise be because of the very polar carboxylic acids, right here and right here, every other unit. Similarly, heparin, which is used to control blood clotting, needs to move freely through our blood, The carboxylate that's on the 6th position of every other unit, and the sulfates on either the 2 or the 6th position make heparin very soluble because they're all charged and very polar. Carbohydrates come into our diets as mono, di, and polysaccharides. Mono and disaccharides are considered simple carbohydrates, largely because they're simple to digest and relatively fast to digest. Complex carbohydrates, like polysaccharides, must be digested into mono and disaccharides when we eat them, which releases carbohydrates into our bloodstream much more slowly and much more consistently. Consistent release of glucose over time is much easier on our bodies, and so dietitians use the glycemic index to indicate how quickly this is likely to happen for each type of food. Things with a high glycemic index tend to release their simple sugars very quickly. Things with a low glycemic index tend to have more polysaccharides, fats, and proteins, which release energy into our bodies over a much longer period of time. Again, slower is typically good.